Okay, here we go. You're ready? So welcome for this first session of the Immersion Project Presentation 2025. The name of this session, constituted by three presentations, is Marine Biotechnology, Physiology and Microbial Systems. We had just before a private session with Matilda Ekholm, and we're going to now following up with Gabriela Diaz, and we'll finish with Emily Hamilton. The jury for these presentations is constituted by on-site Kayla Plichon. Hello, thank you for being here. Paola Furla, online Juliette Armeni for the first presentation. I will take over for the two following ones. Nice. Gabriela, I'm sure you have 15 minutes. So all the students will have 15 minutes, and I will follow with 20 minutes of questions. And you can ask questions also at the end, and online, if some people want to ask questions, they can also formulate their questions on the YouTube Live. We may share them if we have time. Gabriela, the floor is yours.
Thank you. All right, so if you've ever had a strep throat or an ear infection or something similar, you might have been prescribed a medicine with penicillin. Penicillin comes from a fungus that can come from the ocean. This is just one of the examples that have gone breakthrough on medical and scientific advancements through the years. And they come by studying how nature adapts to different pathogens or how it survives. The immune system is very benefited from these studies because, especially in the ocean, because of how in the ocean there's so many pressures and different than on land. And since it's so old, all the organisms in there have adapted to live and evolve alongside any pathogens or any bacteria. Thus, it gives them different immunology than for humans. That is important because this is the human immune system. It's very complex, but I want you to take two different images, very important for this part. One, that we have innate immunity and adaptive immunity. Innate immunity is what we're born with. It's the first step to protect you from any pathogens. It takes seconds for it to adapt. And then the adaptive, as it suggests the names, it adapts as we grow. So it's what remembers the different pathogens and how your body can fight them. Another thing that's very important about this is if you can take a step back, you see how complex everything is, all the connections and all the steps that take into account for the immune system to work. This is why we move to cnidarians. Since the human immune system is so complex, we come and we find simple organisms. They're not actually more simple, but in terms of immune system, if you can think of it this way, humans, you're like a fortress, right? And your immune system can consist of a forest, then you have a river with gators protecting it, then you have a drawbridge, then you have another wall with some more soldiers, and then finally you have a locked door with more soldiers. On the other hand, cnidarians are only the fortress and the wall and a bunch of soldiers around it. So it's different, not specifically more simple. They're also evolutionarily different because of what I mentioned, that how they adapted to grow with the pathogens. And the cnidarians, with like anemones, corals, and medusas, have regenerative abilities that can help you with further study, and also codon patterns that help with reproducibility of tests. They are relevant because there is, along with the humans, the innate immune system is present and it's similar, and we have some specific pathways that are the same. And to study them on a cellular level, we need primary cell cultures. Primary cell cultures are, you get the organisms and you remove the cells and you grow them on a petri dish by controlling different temperature and the media and different factors that they have. This is also good because you're able to modify these factors and see how they affect the cells. And there's also removes the need to rely on live animals. For my study, I worked with 3 different types of anemones: Nematocella vectensis, Anemonia viridis, and Exaptasia pallida, that I will refer to as Aptasia. But for the specific cell cultures, I started with Anemonia viridis. So my goal of the project was to create a reliable cell culture of the cells of these anemones, and I focused my protocol on a paper by Ventura et al., which some people might be very, very used to and very informed about. And I first started by developing the protocol. So the experiment, the original protocol based on cutting first, getting only the tentacles of the anemones, cutting around 20 to 50, 15 to 20, sorry, incubated in glycine. This is that since Niger's have stinging cells, they make them relax and are released in cnidocytes. And then you pat them dry, you rinse them with sterile seawater with antibiotics, and then you cut the tentacles into way smaller pieces so that dissociation is easier. So when the pieces are smaller, you put them in the collagenous so to dissociate. Dissociate is when you separate the cells from the tissue. After you centrifuge it to have the cells separated further from the tissue and to have them be able to extract easier, and then you resuspend the cells. And after you plate them into wells. For the very first trial I did, I followed almost exactly this protocol, but since our anemones were smaller than the ones in the original protocol, we did 80 tentacles instead of only 20. And since we had more tissue, we also increased the centrifugation time to 10 minutes instead of only 5. But our first also trial, we did some experiments with it. So we have 4 different varieties. 1A, which focused on the same protocol, but we saw that we could try to get more cells, and we saw a lot of debris. So we tried to centrifugate it more to try to separate the cells more. This didn't give us enough cells, so it was discarded that same day. Then we added more time in collagenous to see if that increased the dissociation, and it did. We got more cells after increasing the time in collagenous, but after we found out that it was reduced viability and the cells were more fragile. And lastly, we added a step of filtration to the very last one. And the cells that have already been in collagenous, we filtered them to try to remove all the mucus and other debris that were in the anemones. Then we did more experiments. Here we have on the second, we increased the time. Instead of 30 minutes, we did 45. And then we liked the filtering, what it did. So we added that step for the rest of our trials. Then the third trial was the same as the second, and the fourth trial, we added a rinsing part before and the very first, when we had the tentacles, just to remove any like algae or anything else that might have been stuck to the anemone on the tank. And we increased again 90 minutes on collagenous because we thought that would give us more cells. Then we found that actually the cells were more fragile with that long in collagenous because it separates them and could be fragile to the cell wall. So we decreased it again to 60 minutes. And well, since we had a lot of contamination, the culture, the medium, we strengthened it and we made it more concentrated. So it had more antibiotics and antifungal. And we thought that if we had more cells, there would be a better chance of survival. Thus, we decreased the number of wells from 8 to 4. And the sixth trial was very similar, but we increased the centrifugation force. And after this, we on the third day after the first creation of the cultures, we did a change of media or passage, and then it was weekly changes. So our results. This is a bit of what we saw in the microscope. The animal cells that we were looking for are the small white ones that you can see here. And then we have, since it's a symbiotic anemone, we also have this big round yellowish brownish cells. And we also have the stinging cells or the cnidocytes. So what we found, I have here on the trial, the number of trials, but it survived until the last well was disposed of. And the cause of disposal, we have some that were not all of the wells were contaminated, thus not all of them had to be disposed. But here we can see the days on which contamination appeared, and just a reminder of what we saw, of what we changed. So here we have on the first trial, you can see that we saw parasites. It lasted the longest, so we had 63 days, which is great. But we did have some parasite contaminations on the third day. And here you can see the parasites were easy to spot because they will move a lot along the cells. But we see it's these little like grayish cells, maybe not that easy to see if they're moving, but. And then the second trial only lasted 4 days because we got fungi contamination. And on the fourth day, all of them were contaminated and we had to throw them away. You can see here the fungi that's not supposed to be there. And then on the third trial, we got yeast infestation. And this one lasted 35 days with 2 different infestations on the cells. So here you can see these little cells that are like this are the yeast. At first, we thought they were dividing it, but no, it was years. And last thing, we got bacteria. 25 days this one lasted, and bacteria are smaller, so maybe they're harder to see here, but they also move away, so that's how you distinguish them. So you can see we got a lot of contamination. And lastly, with the last two, when we added more concentration to the medium, we had to dispose of them because of that's not enough cell viability. And here you can see that there's not a lot of white cells that we look for, only the other ones. Okay, so basically, it was good because we did fulfill most, lasted more than 20 days. And the culture, but there was a lot of variability. So there were maybe some wells, even though they were handled the same, some of them got contaminated, others not. And it was also difficult, especially for like trial 2, which we got early contamination. So we didn't really see how the variables we changed affected on the long term. And as I mentioned, we also saw that collagenous time, if it was too bad, it just made the cells too fragile. So here's the timeline of infestation on the day of the culture, and here's the number of the trials and the different types of contaminations. And from here, it's interesting to see how most of the contaminations happened within the first week, which showed me that maybe the first week was the most vulnerable, and they could have been maybe not with the handling, since not all the wells were contaminated, but all of them were handled the same. But maybe it could have been with tissue dissociation and in that area of the getting of the cells. And it was similar to other studies that say that during the first week, the contamination is the most prone. Then we found cell count. So I wanted to check if the initial wells when the plating, if it affected how much they survived and how much if there was an increase in cells. So here we can see that the only berry that increases a lot is trial 3. Also, I want to note that trial 5 and 6 are not included in this, or 2 because it didn't last a lot. But also trials 5 and 6, the images needed to be able to count the cells were not available, so they're omitted from this. So here we have the trials, and it's interesting because you can see that even though some of the cells started the same, not all of them increased. And it also, we did get growth on trials 1 and 3. So 3 and 1, a little bit of growth, and 3 growth all the time. And this is how the cell clusters look like, that we were able to know that it was actually the cells that we wanted growing because of past literature. I also wanted to see if there was any relationship between how many wells we had and versus how much they lasted. And from what I can tell, not really. There's nothing that stood out for me. And this was interesting because we did find that it was, I could get the cells last for over some time, but there was a lot of variability that it's important to work on. And this is because we have a lot of limitations. So first is that the small samples, I couldn't make any statistical tests because we only had 6 trials. And that since we changed more than one variable at a time, it was hard to know specifically what variable affected the longevity and the viability of the cells. We also had early contaminations in some wells and whole trials that limited also how much we could see about these changes. And the seeding densities were very different to what we saw in the paper. My seedings, most of them started at around 1 million, while the initial paper had like 150,000. But that is also interesting because even after the passages when we had similar densities as that, the cells did not do any better. So it's hard to compare those two since the densities are so different. And also a very obvious one is that not all of the cell counts were available for all the trials and all of the times. So that would be, in the future steps, it would be good to be able to create more statistical tests, to create more replicates and more tests to be able to really go down on what is the important part and what variables affect the longevity and the viability. And also the contamination control, make a protocol or adjust the protocol for contamination control. So it's not as big as a problem because I had 4 of my 6 trials were contaminated. And lastly, we want to have the cells completely isolated. We only want the cell culture with the animal cells without the other two types of cells to be able to really analyze the impact that the different pathogens have and the variability and how they react to different pathogens. And lastly, the main goal it was to adapt the protocol to Aptasia because that has never been done before. This protocol obviously has been done before by the lab, the main goal was to adapt it to Aptasia. So it's important to know that this wasn't it's for a scientific breakthrough that we want. It's very important to start with the small parts. You know, it's not, you cannot have reliable results on a big test such as immunological research if you can't trust the reliability of where you're testing it. So this is why this is so important. And basically, you have to start small to be able to have a large scientific events. I want to thank very much my supervisors, Francois and Rodolphe, and everyone at the CSM that was so supportive. Also, Maris for putting me in contact with the CSM and being so helpful through this time. I also want to take this chance to thank Paola, since her paper was the one that I was used, and her lab protocols were the ones that guided me through this time, and she was also very helpful at the beginning. And thank you all very much for listening.
Thank you. As we ended with you, Kayla, to avoid any frustration, you can start this one.
Thank you for your presentation. I'll start with I don't know if it's different discussion. Well, whatever. To what extent you contribute to the experimental design? You decided to start. How everything was put in place first?
For my methods?
Yeah. Well, when you arrive to the lab, how you decide from where to start?
Of course. So as I mentioned, we focused our protocol on the only lab that had ever done such a cell culture before, and we wanted to start with that. My initial idea was to develop that for Aptasia, but first, to be able to adapt it to Aptasia, I had to get it to work with the same anemone that the protocol was designed for. And then looking for literature, this paper, Ventura et al., was the one that resonated the most with what I wanted to do, and was the protocol that seemed the most feasible on the lab that I went. And then the different modifications that I had were based on what I thought we were a bit experimental. It was since I had to do the trials. I don't know if you got to see the dates, but they were very, very soon after each other. So I couldn't really see, since these cells take weeks to really develop and you see any change. We were experimenting with different ways to make them work. So it was basically going, okay, I see that I put them more in collagenous, I got more cells. I think that would be better for the growth of them and for the reproducibility. So let's add that to see what happens in the long term. So that's basically how we went. We got the base protocol and then we adapted it a bit of what we thought would be best that would give us better results in a such experimental type of way. I don't know if I answered your question.
But then why? I mean, if you already had this protocol that was working on the species, when you wanted, the goal was to adapt it for Aptasia and Nematocella, if I got it correctly, why you didn't try with more plates, more wells, more material, and in parallel for each of them, the protocol that was already established?
If I didn't do the protocol many times, or
no, I mean, you already had a protocol that was working in another lab. Fair. You wanted to try it in your own lab. Fair. But then why you don't try to follow this exact same protocol instead of trying all these different steps that ended up having a lot of contamination and a lot of troubles? Why you didn't try first this protocol in all of the species and build from there?
Yeah. So the problem is that the Aptasia is very limited on the lab. They're very small and hard to grow. So if it doesn't work, there's not unlimited amount. So we wanted to make sure that the protocol worked because it's harder to get the Aptasia. While anemonia, you don't use the whole organism, while Aptasia you do. and you would have to use a lot of them. Well, in the lab, there will only be so many tanks available, and of like maybe 2 or 3 tanks, and the anemones are like this big. So it was hard to not work with it unless you knew 100% that it would work. That's why we started with anemone. And the first time, if you remember, the first trial, we did it exactly the same, but then it got contamination, and at the very beginning, the cells were declining. It only increased at the very end after like a week or 2, and then we had already tried with other methods.
Going to stats, how many wells did you had for each condition? Because so you had 6 different trials, and you had, if I got it correctly, you had 8 wells per condition. For
the first 4, and then the last 2, we had 4 wells.
Okay.
To try and increase the cell density.
Okay. How then why you couldn't apply statistical test to that if you had n equals 8?
Yeah, because it's not really replicates since they're all made the same. It comes from the same batch, so it's not really the replicates, and also because we tested more. The only trial we really tested as different wells was trial 1, because that's the only one that were varied. Then after all the wells, when I did passages and media changes, they were all mixed together and to change the media and then put again back. So it's not really different replicates. It's just the amount of cells per well, basically. But they're all mixed when doing the media change.
Okay.
Leila, we can go to you, Paola.
Thank you.
Yes. Thank you. Thank you very much, Gabriela, for your presentation. It was very clear for me. Even I have some general question about the question at the beginning. And so sorry, because I'm quite expert on your results. So I don't want to really go on the techniques because it's too easy for me. So I try more to understand the questioning and about the immunity, because you say that you need a simplified model to work on immunity in order to understand how does it work, and in humans it's quite complex, and also in mammalians and other metazoans, so you choose to work on cnidarians in order to simplify the story. But I did not understand exactly why using cells from cnidarians will allow you to simplify the models, because you say at the beginning that the complexity is due to the tissue, it's due to the different mechanisms, due to the diversity of the physiology in complex organisms, and so you choose a one that it's more simple. But at the end, you want to work at cellular level.
Yes.
So this is why I do not understand exactly why this choice, and what did you, so the first, and the second is, what kind of differences do you expect about the immunity at cellular level, not at the tissue level or the organism level, but what kind of different do you expect at the cellular level?
Yeah, of course. This, I know from what I'm not an expert on the actual immunology research. That is Francois, and I know what he has explained to me. But basically, on a cellular level, humans, cnidarians, since we have the different pathways, right? And Francois is looking specifically at one pathway of the sting and G-man pathway. So then they want to basically see if certain proteins added to this put the same pathway, and they adapt it, and they basically make it start. That is what they're studying right now, and thus this experiment would help, since humans they're more evolved in a way. It's a wrong word, but they have a different evolution type. We have like more pathways that can also be the ones that lead to the signaling to the specific sting pathway. So basically, by removing those, you're able to, by studying the anemones, you can, one, compare with human cells and anemone cells, with how the pathway interacts with each other with the different steps, and also be able to remove a lot of the other things that can impact this pathway specifically by that human cell. So it's, I don't know if I answered this question. I'm not really an expert in that area. But basically it's removing other factors that might influence this pathway signaling.
Okay, so your rationale is that even at cellular level, the immunity mechanisms will be different in cnidarians and more sample, so it will be easier to work with.
Yeah, exactly.
Okay, okay. So I have two questions about not the protocol, but something that I think could be interesting from your results, that you want to work with immunity, and at the end you have so many contamination that I think that you really work on immunity. So it was a good way to work on that. And each time you add different different contaminants. So did you have some hypothesis about some correlation between your protocols that you change during your internship? You try many things, and you at the end you ended with different contaminants.
Yeah.
So do you try to find a correlation between the protocols and the contaminants that you obtain?
Yeah, well, it was hard to try to find it. I on it while doing the tests, I didn't even notice that since they were so overlapped and it was hard to keep track of what it was a lot. So I didn't even notice that it was one pathogen per type of trial until I started analyzing the data. And it was very interesting to me because it looks made up. It looks like I try to, I found, okay, these are different types of pathogens. Let's put one in each trial. And it was also hard to test. Why? Because of the variables that were changed. And even trials 2 and 3, they were the same, but they got different pathogens. They got infected by different things. So it was hard to see exactly why. My best guess would be that it's, since we use different anemones, something that was already within the anemone or close by, and with the dissociation, cells just got more fragile. Or maybe the fungi was with managing, since all of the wells got infected at similar times, maybe with 1 day difference. So honestly, it's a very good question. That's something that I would love to look into more if possible, but I don't have a clear answer for that.
Okay, thank you. I can go back with some question, but perhaps I leave Kayla to complete.
I have a technical question, maybe. So let's imagine that you replicate everything, and this time everything works. You have cultures at the end of everything.
Amazing.
How you test the influence on the different factors you changed for each trial?
If I got it to work.
Yeah, let's say at the end you have results for every single condition, and you want to test and to know which factor has the biggest trend influencing your output.
Well, first I would have to obviously have different replicates of the same trial and see how much they work. But then I would use maybe are you asking like the specific statistical test that I would use? Maybe a chi-squared, since it's different variables, or I would go for each of them to see if there's an actual
But of course,
on this. Very good question. I haven't thought about it because I don't have enough data, but basically I would go on each of them to see the survival. I would make a gradient first along to see if the timing in collagenous or the density, how it affects, and then I would make like a simple line plot to see the R squared and the p-value. I would basically, that's a very good question. It's, depends exactly on how many variables I have, because here I changed a lot of them, but I would try to make it, yeah, like a gradient to see when it's a good point, and then see if their survival is different from. Yeah, they're basically a gradient. How we did, I remember because we did it with stats with Stan last year. Something similar with comparing the two. Basically, hone down on which are the two numbers that would be the best to the most that I think would be the best situation. Maybe time in collagenous, maybe I know 90 doesn't work, but I know 45 and 60 minutes does work. So maybe change, add in both of them. Because also in between 45 and 60, it's only 15 minutes. So maybe I will compare these two and see which one works best to try to stick to. Yeah.
I think a lot of very interesting questions have been asked there. So maybe coming back on the objective, did you have some timing objectives, like number of days, a couple of weeks, 20 days, maybe?
20 days was
it was the 20 days.
The most goal, obviously. Longer the better.
Why 20 days? Is it because it's a time, it's the length of an experiment, or why 20 days?
Yeah, we just wanted that as a base. Usually this type of tests take around that time, depending on the test, but with 20 days, it could be enough time to conduct one of the experiments. So that was like the base. Obviously, the idea is to make it longer and hopefully, preserve them in a way.
I smile because I see Paola nodding. And coming back on the contaminations, a lot of questions.
Sorry, Christophe, I just would like to say that it's very frustrating work, because you have, for 20 days, you look at your cells and you cross your fingers, and then you say, okay, I have to go back and to do it. So, okay, just to say that it's not an easy work. So,
yeah, playing with living stuff, and also with microbiology. So contamination is there. I guess it's something you could have expected from the literature.
Yeah.
So being able to compare your contamination with the one on the literature, especially on the same type of species.
Yeah, well, there's different types, and from the literature that I've read, maybe not only like this specific species, because
species of
anemone.
But
different anemone and different corals to face all.
But also not species, but the type of contamination.
Type of contamination.
The timing of the contaminations, it was approximately at the same time.
Yeah. I had this paper that they said that but it was in an anemone, but it was a similar primary cell culture. I think it was in sponges or something. And they had the first week, they said that there was the most fragile one, the most vulnerable, because of the dissociation makes the cells fragile, and also any contaminants that were already in the organism. That they're able to fight when they're in the organism, because it has different methods. Like within the organism, it's easier to fight them. But after you dissociate them and remove them, it's harder for the individual cells to fight these type of pathogens. So it makes sense that most of my contaminations are on
for the bacteria, for example, to use antibiotics against fungi. Yes.
Yeah, so that's something that we try to change. So if you remember on my last 2 trials, we had the concentration of the media. The media had antifungal and antibiotics. So we said, okay, we're getting a lot of contamination. Let's make it stronger. The problem with that is that then the cells weren't really able to handle that. And the cells became too fragile. So we saw a lot of cell death.
Due to what? Due to the
how the amount of antifungal and antibiotics.
Because you just increase the media in general, but
not the concentration of the media.
The concentration.
Yeah, the concentration. The amount of media was.
But media is more than only antibiotics. Maybe you have other stuff you also. Yeah. Have you tried to only increase antibiotics and not the other?
No, I didn't try that. No.
And do you think it could?
I don't think so, because the problem is that these are harmful to other contaminants and like parasites and bacteria and fungi, but they can also be damaging to the specific cells. From what I've seen and what I've read, too much can make the cells fragile.
Thank you. 2 minutes for questions from you guys. Allez. Yes. Hilda.
No, just curious. It's a complex topic, and also maybe what Paula said, it's frustrating. So what do you think was the most challenging thing during your internship when working on this?
Figuring out why the cells weren't doing good.
Okay.
That was hard, and also the time, since we had a lot of time limitations, and therefore we had to overlap a lot of them. And we didn't know in the long term if what we changed on the first one actually did any benefits on what we did. Like for 1C, we added time in collagenase, and we saw, oh wow, we see a lot of animal cells. This should be very good. So then we kept increasing collagenase. But in the long term, that was not beneficial for the cell. So it was hard to work on top of another and another without really knowing what the long-term effect on the cells was.
Yeah.
And the separation, since we had a lot of also working with the other two types of cells, it was hard to really specifically know what was good. It was, and it was also confusing, like, because even though we thought cells were dividing more than once, and last time we saw something very interesting, that the algal cells were not green anymore. So they had left the culture. So that, I think, would also be interesting for a study, but also that made us think that it was good. The cells were dividing, but the cells didn't divide in that one. It was the algal cells that something was happening there. But yeah, that was a challenge.
Yes, Salma.
And when did you first realize there was contamination, and how did you analyze under microscope found cells?
Yeah, it was on most of the days it was day 3. So before we tried to, we checked the cells every day, but it was, we saw, because how different they look. We already were used to what the cells looked like, and we saw like the parasites and bacteria moved. So we could see that clearly the cells weren't supposed to move. So then if you saw something swimming around, you saw that was contamination. And at first I was scared. I didn't know what it was, but then my supervisor was like, yeah, if they move, it's that. And then the fungi was obvious because you could see them on the top of the well. You could see it wasn't right. And then the yeast, at first we thought that the cells were dividing, but then yeast had this specific shape. So that's how you could tell that it was yeast and it was a contamination.
Last question.
So is it possible that having the contamination could lead to then have unexpected but good results, meaning that the cells somehow react?
Yeah, actually, on the it was right before I left, so I'm not sure how it ended. But when I was leaving, we had a well that was contaminated with yeast. Yeah, yeast or parasites, if I'm not mistaken. But I wasn't in charge of that trial anymore. But there was a yeast contamination, but apparently the cells were doing really good. So they kept working on that. Couldn't tell you how it ended on if there was actually a good reaction or just the cells developing some that did not mind about the yeast and they were able to continue. But yeah, it's possible.
We have a last question from online. I will say it to you. So do you think cryptic species in the cnidarians you use could bias immune responses you want to explore? Cryptic species. Do you think you could have cryptic species in your culture?
Cryptic species.
Cryptic species.
Sorry, I'm not. What is cryptic? Like,
So basically, you have the same morphology of the animal. You think it's a one species, but in reality there are multiple species.
okay, like,
From the same morphological shape. The same morphology,
meaning on the anemones or the contamination?
I guess the anemone.
In the cnidarians.
Yeah, I don't see why not. I think that it depends. Like, I know that in the lab that I worked with, the anemones that I worked were all the same species because it's been studied. There were ones that my supervisor had been growing and everything, but I don't see why not. And I think since the organisms would be very similar to each other, probably the results will also be very similar. depending on what, but honestly, something that I'm not really
Yeah,
familiar with.
you can say that, you know, it's
Yeah,
totally okay to.
It's not something, but that would be my best guess that it would behave kind of similarly, which is kind of what we are depending on since we want to use the same protocol on a completely different species.
Okay, let's conclude on that. Thank you, Gabriela. So we'll conclude this first session with Emily Hamilton.
Where am I right?
Yes, we're just closing the door so you're not annoyed by anything. The floor is yours, Emily.
When you first look at this picture, what is the first symbiosis that comes to mind? If you said zooxanthellae, you would be like a lot of people, including ChatGPT, who said the same thing. However, this study is not about zooxanthellae. It's about another component of the coral microbiome. This study is about the protocol optimization for and preliminary analysis of microbial bacterial communities in Physiologous species in BIOTECHNOLOGY, Costa Rica. You may already know that a coral is of the phylum Nidaria and that they have diverse morphologies. You may also know that they are nicknamed the rainforests of the ocean and that they host 1/4 of the world's species. However, did you know from this 1/4 of the world's species, this is also included of what's inside the actual coral? This is called the coral holobiont, and it is the complex assemblage between the coral animal and their microbial partners. Coral health is dependent on the microbial stability and it can underpin both reef functioning and marine biodiversity. In this coral holobiont, this can include the zooxanthellae, which I've already mentioned, eukaryotes, bacteria, archaea, as well as viruses. They may live in or on the coral tissues, on the surface mucus layer, within the gastrovascular cavity, as well as in the skeleton. They may contribute to functions such as coral growth, genomic modifications, and metabolic support. Now, in contrast to the zooxanthellae, which is extremely studied and well characterized, other organisms in their microbiome remain less well characterized. And the research is limited to the most common and identifiable members, which may limit the conclusion of potential function and identification within the sex. The microbiome composition can be influenced by many biotic and abiotic factors, as well as can vary across large geographical regions within the same area, however, upon different coral reefs, and actually at different depths within the same reef. This is why regional studies are very important. However, why study the coral microbiome? Understanding coral reef functioning and how they potentially can adapt, change to in different environmental conditions can improve understanding of how microbial communities influence coral health and resilience. This, in turn, can predict bleaching outcomes and help enhance restoration efforts. So this leads me to my question. What are the bacterial communities present in Physiologous of BIOTECHNOLOGY? This will be developed through two objectives. The first one is to develop and standardize a protocol for the sampling, preservation, and extraction of bacterial DNA, as well as provide a preliminary characterization of bacterial communities. However, to begin, where is BIOTECHNOLOGY? It's in the North Pacific of Costa Rica, right here in an area called the Gulf of Papagayo. It is a socially economic area and has some of the most developed coral reefs in Costa Rica. However, over the last 2 decades, there has been a large decline in live coral coverage, which makes it a prime target for coral restoration efforts. If you look at the species Physiologous, it is the main species in their coral restoration efforts, which is why it has been the coral study within the study. So it was collected by scuba diving in 3 sampling events from March to May. 10 colonies were selected and tagged. Then there was pull-up extraction and DNA extraction, which, when successful, was sent to sequencing in Korea. So, as I just mentioned, the protocol started with field collection. And then we tested 2 pull-up extractions, airbrush and bailout, to see which one was the most optimal. The first one, airbrush, is when the coral is sprayed with sterile seawater at a light pressure to remove the coral tissue and mucus from the skeleton. And the second one, bailout, is an adapted protocol which actually focuses on polyp bailout, which is a stress mechanism that bacilloporid corals experience. This utilizes evaporation-driven hyperosmotic treatment, which I'll get into a little bit later. And then 2 DNA extraction kits were utilized, DNA Easy and Powerlyzer. The difference between the kits relies on the cell lysis mechanisms. DNA blood and tissue kit, or as I call it, DNA Easy, uses an enzymatic digestion to lyse the cells, while DNA Easy Powerlyzer Power Soil kit, or Power Soil, contains both mechanical digestion and enzymatic digestion, as well as it contains inhibitor removal technology, which again, I'll get into a little bit later. And it's important to note that both were executed with the manufacturer's protocols. So the airbrush method was utilized in both field conditions, so that meant directly after sampling, and in laboratory conditions. However, the bailout method was only done in March under laboratory conditions, meaning the coral samples are transferred back to the host laboratory in the cavity. The DNA extraction success was evaluated using 2 main indicators: the DNA concentration and quality, which was analyzed using a nanodrop. DNA quality was considered of high quality when it was between 1.8 and 2.0, and then this was further evaluated using gel electrophoresis. And bacterial presence was also used as an indicator using polymerase chain reaction, and then again verified using gel electrophoresis. The primer used was 16S RNA gene, which is the RNA component of the 30S subunit of the prokaryotic ribosome, which is a great primer because it's a highly conserved sequence in bacterial species. So overall, across all the DNA extracts, there was a DNA concentration of 30 nanograms per microliter and a mean DNA purity of 1.6. It is also important to note that all the DNA extracts appeared on the gel electrophoresis. However, when it came to the PCR, only 55% of the extracts amplified, which means only 55% appeared to have bacterial DNA. It is also important to note that no APRO samples produced a detectable PCR amplification, which means that none of them appeared to have bacterial DNA. When looking at the 2 polyp extractions, the airbrush method and the bailout method, as you can see in the orange, in the bailout method, it appeared to have a higher DNA concentration as well as mean DNA quality. However, none of these results were statistically significant. So we went to look at the gels. From the electrophoresis gel, as you can see, the bailout method produced very concentrated bands when being compared to the airbrush method. However, the airbrush method did produce bands. Although, why did we prefer to use the airbrush method, and why will they continue to be used in the standardized protocol moving forward? And this can be due to some of the impracticalities of the bailout method. So in the bailout method, coral fragments are placed in seawater and then placed in a dark room in an open Petri dish for 24 to 48 hours. This allows for the water to evaporate, increasing the salinity, hence causing hyperosmotic stress. This resting time can increase contamination risk because it's an open Petri dish in an open area, as well as makes it unsuitable for field protocols. In addition, this resting period may actually change the composition of the bacterial community. When looking at the airbrush method, it is more practical during field sampling because it can be done directly after sampling. It makes it therefore easier to standardize, reducing contamination risk and reducing the chance that microbiome has a composition change. Many of these protocols are already established, and actually making the airbrush method a standardized protocol, which is why it was the selected method moving forward. And then looking at the two DNA extraction kits, as you can see, the PowerSil kit had a higher concentration of DNA and DNA quality. And this time it was actually significantly significant. However, why did PowerSil lead to higher DNA? And this can actually, the performance difference can actually be accounted for the different chemistry and lysis mechanisms of the kits. Lysis mechanisms can actually produce a bias because they can actually have different affinities for specific cell distribution. So, for example, some lysis mechanisms may favor eukaryotic DNA or oral DNA over microbial DNA. This means that when doing the sequencing results, that the apparent community profile may be shifted. When looking at DNA-EASY, even though it has been successfully used in microbiome studies, it focuses solely on enzymatic digestion using proteinase K, which may shift the community composition more to eukaryotic DNA. In addition, this kit has also been optimized for eukaryotic tissue DNA. When looking at PowerSoil, this actually combines mechanical disruption using bead beating with enzymatic digestion, which may favor a larger variety of organisms. And in addition, this protocol has been optimized for complex environmental matrices. Secondly, coral tissues contain a lot of PCR inhibitors, which such as humic acids, due to residual skeletal tissue. This can cause this may impact PCR efficiency as well. It may create false negatives. The PowerShell kit includes a step to remove the PCR inhibitors, which is not present in DNA-EASY. This is consistent with some of the field restraints that we have, the laboratory constraints that we had within our study, as we had no amplifications with the aerial samples, which used the DNA-EASY kit. Lastly, during field work, there was practical restraints using the DNA-EASY kit. This kit requires high centrifugal forces to remove the wash buffers as well as dry the column, and the centrifuge that we had on the field did not reach these forces. This can contribute to a lower ratio as well as cause PCR inefficiencies. PowerSolid is also commonly used for DNA extractions from coral biomass and has been standardized in these protocols. So overall, this is the protocol that has been standardized. It starts with field collection, then will be the polyps will be extracted using the airbrush, and then the DNA extraction will be using the Power Slicer kit. This protocol was then used to send 3 samples to Korea for amplification and sequencing of the 16S rRNA gene. However, of the 3 samples sent, only 1 out of 3 samples amplified. So why was that? Well, it could have contributed to DNA degradation because there's a lot of moving of the DNA of the samples from on the field back to the host laboratory all the way to Korea, which could have created suboptimal conditions for DNA preservation, as well as there was not enough bacterial DNA. The sequencing, we also sequenced using an ITS primer, which they were testing for fungal DNA, and all of those were successful. When looking at the gel, the successful DNA extract had the thickest band, and the other samples, there was a band, but they weren't as thick. In the future, we could concentrate the DNA further or use less of an extraction, the dilution buffer, when extracting the DNA from the column in the kits. However, we still had one sequence which produced some results. There was 153 amplicon sequence reads, which means different sequences that were identified. Of this, there was 36 taxa. The most abundant taxa phylums were Proteobacteria, Firmicutes, and Verochromicrobiota. The top two phyla, Proteobacteria and Firmicutes, actually is coherent with literature as the top 2 phyla present in Bacillopora and many of the databases. And this demonstrates that there is a broad phylum level consistency across coral holobionts, and this may be due to the functional importance of these phylums. Proteobacteria is important for holobiont nutrient acquisition, nitrogen acquisition and cycling, and Phenomycutes is as well important for nitrogen inputs through nitrogen fixation, and it actually produces metabolites such as organic acids and phosphatase enzymes, and they make them into bioavailable forms for the reef and the actual coral species. Nitrogen is important for the reef because it is frequently limiting, and it is supplied to corals through symbiont uptake or host heterotrophy. So this leads me to my limitations of objective 1, which I split them up into different objectives. So in the first objective, several steps occurred under field conditions, which I know said reduces a risk of contamination, but also can increase it because the environment was not a sterile environment like in the laboratory. In addition, only using DNA concentration and purity and PCR amplification as indicators may result in some bias. As you saw, the sequences that came back, only one of them worked. So therefore, sequencing should be used as an indicator in the future, as well as there was a small sample size, which reduces statistical power. This is also a limitation in objective 2 because we only had one sequence that worked. So therefore, this is only at one time point at one location on the reef, so therefore none of the communities can actually be concluded. As well as none of the there cannot be a conclusion on any functional profiles until a punitive metabolic test is done. So, in conclusion, the aim of the study was to characterize the bacterial microbiome of Bacillopora in BIOTECHNOLOGY, Costa Rica. We had 2 objectives. The first objective was that a protocol was standardized, and this standardization included using airbrush and the Power Soil DNA kit, as well as objective 2, which was to create a pilot characterization of the Bacillopora microbiome. The microbiome is important because its understanding of how microbial communities influence coral health and reliance can help predict bleaching outcomes, which in turn can enhance restoration efforts. So, thank you.
Okay, here we go. We can go for questions now. I can reopen the floor to Paola. Yes, Kayla, you okay?
A bunch of questions.
Bunch of questions? Well, may have a couple. Paola, you can start.
I can start. So again, another very well presented defense. I think that you did what we really expect as a master defense. So really, you did the job. Really, it's what I expect. I don't want to say that the previous one was not, but that there are more scientifically, I would say, those kind of presentation in seminars. Yours is really a master presentation, and you try to understand all the different steps of your protocols, and you try to give some arguments, some support to all your steps, and I really want to congratulate you about that. So good job. Then obviously I have some questions. First question, it was first question about the objectives. You say that to understand the microbiome will help for restoration methods, and I really don't catch the point. How knowing the bacteria you can help restoration?
So the Universidad de Costa Rica, they do a lot of restoration efforts. So they like to combine scientific-based approach with like more specific microbiome genetics with ecological-based approach. So they have other projects that are testing feasibility of different coral gardening methods. However, if they can analyze the coral microbiome, because it can actually shift depending on bleaching, different environmental conditions. Therefore, in the future, if they can fully grasp when the microbiome would shift, they can predict when there would be the bleaching outcomes. And this, in the future, this study will go to comparing bleaching versus non-bleached corals. Therefore, if they were to think that that there would be a bleaching in the future, they could start to look at the microbiome. However, I do understand that sometimes sequencing can take a long time to come back. Therefore,
Yeah.
the bleaching event may have already occurred by the time that the
What you're saying for me, it's more monitoring than restoration. Yes, for sure, the bacterial shifting could be a signal of symbiosis impairment or something that they tell you that something goes wrong. But it's more for monitoring.
yeah,
I
would
was
say-
thinking, yeah, please add what you want.
oh, sorry, I would agree with you as well.
About the restoration, what could be interesting is to try to correlate some kind of bacterial signature with some healthy state in order to say, okay, if I have to choose some colonies for transplantation and restoration, I will prefer to choose the colony that will have this kind of bacterial signature, like a prediction of the resistance of the colony. In that way, I would say yes. But on another way around, you say that you prefer to use the airbrush than the bailout because as soon as you take the sample and you put it on your devices, you could shift the bacterial communities.
Yes, exactly.
So it seems to me, but it's not a criticism. It's more discussion. It's to see, okay, if this bacteria is so flexible, so shifting rapidly, is it really the good way to tackle the coral resistance or the coral efficiency for restoration? What do you think about that? I try to push you in my direction, but perhaps you
Yeah.
have some other arguments.
I do agree because it can rapidly shift between different states. However, I think more research has to go in on how rapidly the actual microbiome can shift because there's a constant flow with the seawater, with the sediment. Therefore, a lot of the bacteria inside the microbiome can be contributed to just inflows from bacteria from seawater, which I found in some of the analysis of the different families. And however, I did find some literature that said Bacillopora has a relatively stable microbiome, which is why it can be used as a good indicator.
Okay, good answer.
We can go to Kayla. Oh, yeah. Maybe a last one, or we can go to Kayla and come back to you. We'll have time as well. We can go to Kayla.
So congrats on your presentation. It was very clear and easy to follow. I have a bunch of questions, but I tried to select a few of them. So for technical questions, I got a little bit lost. So you sampled 10 colonies per sampling time.
Yeah.
Okay. What was your N for your replicates for each procedure you did?
So we so the first in the March, we sampled 10 colonies and tagged them so we could come back to the same corals. And then the N for all the DNA concentration was all of the months put together.
All the months put together.
So I made an overview of all the DNA concentrations and quantities that I got, which was the N. So it was across all 3 months.
And so, can you go back to your slide of the methodology, please?
Here.
If you have one that shows the timing and also how you use the samples.
Oh yes, I do have how it's in my annex.
Because I kind of lost with them. How you use it.
So I had to compress the files, therefore it's a little bit blurry. But essentially in March, we split up the corals into 2 different sections. So some of them we actually, to try using the same coral, we took some fragments and did airbrush with them. And this is the same fragments from the same coral, but different fragments with the bailout, because essentially they would have the same genomic DNA give or take.
Okay.
Yeah, so that's how I created the end, was that I added up all of the first row.
Which added up all this first row. Okay. And do you think because of doing that, would you have a time effect on your results?
Yes.
And how can you control about it? Because from what I see on your experimental design, you have a bunch of factors that could influence your output. How would you control for that?
So for the differences between the mechanisms, I only actually used the March sampling because that was the only time that we used both different polyp extraction methods. Therefore, I only calculated using those, I only calculated using the March sampling, which is why I think it also contributed to the insignificant result, because it had a lower end value.
Okay. Thank you. So you talked also that you found some bacteria or microbes, anyway, microbes that are present in the column, in the seawater column, that in your actual extraction. How do you control for? Did you ever control for seawater influence or even the substrate?
Yeah, so in the last sampling, we collected sediment besides the corals, and we tried to do the DNA extraction from there. However, it wasn't successful because I think there was just not the optimized kits for it, as the sediment is very thick. It didn't go through the DNA columns very well. Okay. Therefore, I think another method would have to be used for that.
Okay, but then it wouldn't make sense to use the same method for the extraction of the surroundings and in the colony? Or do you think it wouldn't change anything?
I don't think it would change anything.
I'm trying to go through my questions.
I have one issue too.
Yeah.
I'm trying to choose the questions.
Paula, go ahead.
A question about the techniques about your DNA and how you analyze the DNA and your criteria to identify which one will be the better. You use the amount of DNA that you recover after your extraction and also the ratio from protein and DNA. Is there any other criteria you can use before sending the samples?
We also used gel electrophoresis. So before actually doing PCR, we ran it through a gel to see how the DNA looked and if they all look the same compared to a control.
And what does this gel inform you about?
It informs if it's of good quality, it will run very clean. There'll be a nice, how do I say it? It'd be a nice, sorry, I can only think of the French word. Like it looks very nice.
What is a nice DNA?
I should have put a photo in. It essentially, at the top, the DNA appears to be very homogeneous. Therefore, you can use the gel to see the different sizes, as smaller DNA will travel quicker than the larger DNA samples. Therefore, you can see the different DNA sizes. So theoretically, it should be a nice bar that travels down straight and parallel to each other. However, if you have different sizes, if you only, for example, extracted smaller sized DNA, then most of the DNA will be concentrated at the top of the gel versus at the bottom.
Yeah. Yes and no. It's better to have all the DNA at the top.
Yeah.
Because even the bacteria DNA, if it's shorter, sure, it will stay at the top of your gel. So if you start to have this line, as you say, this homogeneous line, that means that you have degradation. And in your gel, it was not so easy to see because on one line you have so many DNA that for sure you can see degradation because it's very overloaded. On other lines, there is a few amount of DNA, so we couldn't see any degradation because there is a low signaling. What I mean that perhaps the gel that you present to us today cannot really help us to say, okay, this is a good line to choose. Because as it had not been loaded with the same amount, it's difficult to compare about something that homogeneous, something that it's really on the top, or something that it's totally degraded. Well, the totally degraded, yes, we could have seen it, but not with the other condition.
In the PCR protocol, we the DNA concentration that was used was between 15 microliters and 20 microliters. So we did standardize for that.
Yeah, it's a concentration.
Mhm.
Concentration, volumes, or quantity?
Concentration, 15 nanograms per microliter. 15 to 20.
I prefer.
As we're talking about DNA and stuff like that, let's come back to questions really amateur. Right. If you come back on slide 41, sorry, you have to go all the way back to that. But you're talking about a ratio between A to 60, A to 80. And once again, you were talking about nice DNA here, you're talking about good quality of DNA. I'm not familiar with that. So what about this ratio and why is it a good indicator of quality?
It's the ratio between, I didn't know this, wait, between DNA and protein. Therefore, it means that if you have a ratio between 1.8 and 2, it tends to be it's a good ratio between the two.
So it means that 8 to 60 is what, the DNA and the other is a protein?
Yeah, or it could be vice versa.
Okay, so it means that when you have 2 times more DNA than protein?
Yeah, then it's a good indicator. However, if you have a lot more DNA, there could be other, when it's too low, there could be external variables inside your DNA. It could be more impure. And then if it's too high, it also means the same thing.
A few things also I haven't got. It's when you talk about the bailout methods, you say that it's not compatible with field protocol. Once again, I'm not familiar with that. As I understand, you have like a petri dish, you have corals, you wait in the dark. And how is this not compatible with field?
Because it has to be in the dark in one space for 24 to 48 hours. So if, for example, if you're on the field only for a day.
When you say on the field, it's like on the vessel,
On the boat, yeah.
On the boat, yes.
On the boat, it's harder to be able to get it to stable conditions.
Okay. If it's like on Tahar, for example, for multiple days, it could work.
Yeah.
Okay.
For our protocol.
Your protocol for your field work.
Yeah, exactly.
Okay. And also last one, before I give back the floor to Kayla, I'm sure I found out more questions. I really appreciate at the end you're addressing ecological functions of the biota. Interesting. You mentioned nitrogen acquisition mostly. Are you aware of other ecological functions that this biota offer to the coral community?
Yeah, the specific proteobacteria or just in general.
Because more generally, if we come back more generally to your approach, you'd say, okay, you have an inter-specific relationship we know quite well with horizontally, but you unzoom and say, oh, there is much more. And how much this much more variety of species living together provides function?
So everything that I say has to take lightly because we didn't do a putative metabolic test. So all of this is based on past literature that I read and their function from putative metabolic tests done in Faciliflora, however, in different regions. I'm gonna go. The Bacillus, which is a species, which is a genera of Firmicutes. There's a lot more functions outside of just the nitrogen acquisition and the creation of organic and the secondary metabolites, as it can also be an indicator of coral health, as it can, this one's actually, there's some research, there's some literature that actually said if it's there, it means that it's good, it provides to the immune system for a specific type of pathogen. However, there's also some literature saying when it's there, that means that there's pathogens in the water. So therefore, it means that the coral is not in a good state.
Yeah, but it can, whatever they're pathogen or not, it's not, I don't know, maybe more, but they can provide immune system.
Yeah, immunity response, and they actually are in symbiosis with the zooxanthellae as well, and that contributes to the nitrogen fixation and immune function.
And is there any kind of predation? I mean, predation like can that be kind of food as well?
For the coral,
Can that be processed by the coral?
Sorry, I don't think I understand you.
If it can be a source of food for the animals, for the polyp itself.
Oh, like as almost like the zooxanthellae.
Yeah.
From all the literature reviews that I've done, they tend to do another function because they already have the zooxanthellae, which provide a lot of the organic carbon needed to do cellular respiration in the coral.
Back to Kayla, maybe for last question.
Maybe jumping on one of your questions was, so you were saying that if we observe the presence of a certain microbe in the colonies, it's an indicator of health. How do you assess the health of a coral, and if it's actually due to the presence of this microbe, or if it has nothing to do with it?
It's this one, it was hard to do because we only had one sample. However, in other literature that I read is that they could.
What would you do?
What would I do? I would compare a coral that visually appeared to be covered in algae or be bleached versus a coral that visually appeared healthy. However,
What do you mean visually?
there's, for example, there's no algae covering it. There's a good color.
What's the gold color?
For Bacillopora, it's an orangey red.
Depends on the species of the Bacillopora.
Yeah.
Because
For this, the Bacillopora in the BIOTECHNOLOGY region.
which species is it?
They actually, it's between, this is why I just said Bacillopora species, because they have two species. It's Bacillopora Darnicus and Bacillopora verrucosus. However, they haven't actually done the, they don't do the genetic differences because they're both present. Therefore, so therefore they don't differentiate between the corals because morphologically they look very similar. And since it's just for restoration efforts, the difference between the corals is they don't believe matter as much.
But we're going a little bit more into the technical part. How do you measure the good color of the coral? Because when you're comparing to a bleached one, I guess you're trying to see if it's in symbiosis with the zooxanthellae compared to the bleached one. But can you do actually to measure it?
When comparing to a bleached coral, one thing that I didn't say was that when we did the coral extractions, we verified under the microscope that there was some, like the zooxanthellae and other bacteria present in our sample.
One or two or versus?
We just looked just to make sure that they were there. However, I think in the future they could look at more of the concentration and compare the different concentrations of the amount of zooxanthellae inside the corals.
Yeah, so we can open the floor for a question from Alex.
Yeah, so the idea, it seems like, was to create an optimized method. And I was curious if you think that this is something that could be used in the region by other scientists, or do you think that this will contribute to some of the methods that people use in the future?
Yeah, so the project that I was working on is part of a 2-year project. So since I came in right at the start, they didn't have a protocol for the actual microbiome extraction. Therefore, what my job was in the first 4 months of the project was to figure out how they could do it. So in the future, this protocol will be, there is actually their standardized protocol that they'll use for the microbiome extraction.
I was wondering, because my knowledge here is very limited, but you had to take the corals from, I guess, the reef and to analyze them. Are there any ways to analyze the genetic contents without taking the actual animal? Could you, I don't know, harvest the microbiomes?
The
in the field?
calcium carbonate skeleton makes it difficult because some of the methods actually include crushing the coral.
Okay.
And you run that through the tube. However, because it doesn't degrade very well, it sometimes results, when we tried it resulted in low DNA quality and quantity, especially quantity, which makes it hard to do downstream processing.
Last question from Ron.
I was just curious if this type of DNA extraction is costly and if you think it's the most strategic use of funds for this program.
That's it. I know for DNA extraction methods, using kits tends to be a little bit more expensive because for a kit to do 50 to 100 extractions, it can cost up to 500, 600, 700 euros. And for the actual polyp extraction, since we're only using an airbrush and sterile seawater and sterile plastic bags, that is not very costly. However, the DNA extraction can be costly, as well as sending the DNA off for sequencing, which I know that we had to wait for there to be 10 samples before we could send it off, because if not, the price didn't match the amount of samples that we're going to send.
Thank you. Thank you for your questions and answer. I have someone online saying also congrats. And it's now time for a coffee break. So we are coming back very soon.